Hi everyone, welcome to the lesson primer for stats in AP Biology. Some of you may have already taken stats or you'll take stats in the future, but the way that we do statistics in AP Biology is slightly different. We conform to the way that statistics is done for science and science reporting, so I'm going to go through with this with you all. So the first part of statistics that we're going to learn about is standard deviation. So this is the most widely used measure of variability. Essentially, it is the average of the distance between each measurement point in the sample and the mean. So the larger the spread of data, the larger the standard deviation. And so it's basically measuring the precision of your data points. So let's look at an example. We have two test averages for teachers. Many times students are freaking out about, is the test hard? Well, when a teacher says that the average is an 80, that can mean one of two things. That can mean that in this case, for example, the average is 80, but all the dots, all the grade points are pretty close together. So nobody ended up failing, nobody got above a 90, but all of the grades are somewhere close to the mean, which is an 80. Now let's take a look at the second teacher. So the second teacher also had an average of an 80, but notice the difference between, here is the average, that's the 80. Look at the difference in the test scores. We had some students that basically failed, and we had some students that did extremely well. So when you average all of these out, it still comes out to an 80. So in science, there's got to be a way to understand what this average means. And that is what standard deviation is. So when you look at how does the mean for teacher 1 compare for teacher 2, the mean is exactly the same. The two figures show what kind of data was gathered for both teachers. How would you describe the distribution of test scores for both teachers? And keep in mind, what we mean by distribution is where are all the grades sitting? So even though they had identically the same average, you can see that here, all the grades for the students are very close to an 80. Whereas this teacher, you had some that are extremely low and some that are extremely high. So question number 3, which data set has the larger standard deviation? Remember that we said the larger the spread of data is, the larger the standard deviation. So that means that teacher 2 data would have the larger standard deviation because the data points are very spread out. Whereas for teacher 1, the data points are very close together. The second part that we're going to talk about is standard error of the mean. So standard error of the mean bars are helpful in determining if there's a statistically significant difference between two sets of data. For example, a control and a test group. Typically in biology, data will be represented with a bar graph, and then SEM bars will be drawn as shown in figure 3. So this is a very important concept. And many times students will lose points because they forget to say statistically significant. So we're going to practice with this a lot this entire year. So don't worry if you don't get it right away. We're going to practice and practice and practice and make sure that by the end of the year you understand when to write statistically significant and when not to write it. So let's look at a practice graph. We have two sets of data here, two graphs, and you'll notice that they're bar graphs here. And the SEM bars are these lines right here. These are what are known as standard error of the mean. And you don't need to know how to calculate standard error of the mean, but you're going to learn how to draw them and how to interpret them. Now, if you ever take a statistics class, you will be drawing or calculating them. But in AP Biology, we calculate it already for you. So again, your regular bar graph, just like you've seen since, I don't know, probably second grade or whenever. And these are what we're adding new. These are called SEM bars. And they're basically telling you that the true average, because we can't test every single data point that ever existed, the true average of your data lies somewhere in this range. So question number 1 says, in which set do the SEM bars appear to overlap? So let's just arbitrarily call this one set A and set B. And we're looking to see when the SEM bars overlap. So in other words, when the top of this SEM bar would hit the same value as the bottom of this SEM bar. So you can see here that the top one doesn't actually touch the bottom of this SEM bar. Whereas this one over here, you have the top of this SEM bar and the bottom of this SEM bar. There's overlap in this range. So right here in the yellow area, this is where the SEM bars overlap. And what that means is that the true average of this data could be as high as this, but it could also be as low as this. Whereas the true average for this data could be as high as this and as low as this. And so since there's overlap here, that is something to take note of. So if the SEM bars overlap, we cannot say that there's a statistically significant difference between the two sets of data. If the SEM bars do not overlap, we can say that there is a statistical difference between the two sets of data. So in which data set of figure 3 can we say there's a statistically significant difference? That would be in set A. So again, for me, I think it helps to think about these SEM bars as a way to see, does my information change or does my conclusion change when I look at these data points based on the SEM bars? So here's an example. So let's say that you were looking at data for a graph, a bar graph, just the plain bar graphs like you've seen before. And you were comparing group 1 to group 2. Well, when you compare these two, it looks like 2 is higher than 1, but there is a difference between them, and 2 is bigger or has more than 1 does. Now let's add the SEM bars. And again, you won't have to calculate the SEM bars. That number will be given to you, and I'll show you how to add those two bar graphs. So what we want to see now is if I redraw these SEM bars or redraw these bars based on the height of the SEM bar, I want to see, does it make a difference in my data or in the conclusion that I draw? So we originally had said that 2 was higher than 1. Well, let me redraw my graphs now. So I'm going to draw graph number 1 at the very, very top of the SEM bar. Okay, so this is now graph number 1 and what the new graph looks like. Now for graph number 2, I'm going to draw it all the way to the bottom of the SEM bar. So I'm going to take my eraser and I'm going to draw number 2 here as low as that SEM bar will go. Okay, so there's my new graphs. Now if you were looking at data like this, you cannot still use the same conclusion that 2 is bigger than 1, because now 1 looks like it's bigger than 2. And so with this information, that tells us because these can flip-flop and my conclusion changes, there's actually no statistically significant difference between these two data points or between these two data sets. And so using statistics can help us understand a lot more about the data and what's going on with it. Now let's do some guided practice. An AP biology teacher is looking at AP scores for the previous year and compares them to how students did on their knowledge check averages. In other words, their daily averages versus their AP scores. And we use total points, and so these numbers may look small, but 18 out of 20 is a 90, 16 out of 20 is an 80, and 14 out of 20 is a 70. So don't worry about the percentages. We're going to just graph the numbers here. So on the axis provided, we're going to create an appropriately labeled graph to illustrate the averages of the 3 groups and then draw the SEM bars in. On your graph, write the min and max values for your SEM bars. So let's go ahead and do this. The first thing I'm going to do is create my categories. So this is going to be students who got a 5, students who got a 4, and students who got a 3. That's going to be my x-axis. Along the y-axis is going to be their average. So the highest number I'm going to need is going to go all the way up to 18. And there are, I believe there's 20 boxes here. So I'm just going to count each of these boxes as 1 all the way up to 20. Okay, so now what I'm going to do is I've labeled both my axes, or I've numbered both my axes. So this is the daily score. And then this is the AP score down here at the bottom. I'm going to start with students who had a 5. I think the easiest way to do this is to graph this just like a regular bar graph. So if students who got a 5 averaged a 18 out of 20, then what I'm going to do is I'm going to take this data and I'm going to go 15, 16, 17, 18. I'm going to just go ahead and draw my bar graph here for the 5. I think that's the best way to start. Now let's add the sum bars. So for the sum bars, what I'm going to do is I'm going to take my average and I'm going to go plus and minus this amount. So let's start by going up. I want to do plus 0.89. So from 18, that means, and I'll do this in a different color so it's a little bit easier to see, that my sum bar, when I draw it, is going to go up to 18.89. And you can see I'm very shaky here. And so what I like to do is I tell students, write down what the top of that sum bar is next to it so that there's no doubt in anybody's mind as to how far up that goes. So this is 18.89 is the top of my sum bar. Now to get the bottom of the sum bar, I'm going to subtract 0.89 from 18. And we can use calculators for this part. So when I enter that in, I get 17.11. So about right there. And again, I'm really worried that because I'm, shaky with the iPad, I'm going to go ahead and write down 17.11 on my data. Okay, so 18.89 was 18 plus 0.89. 17.11 was 18 minus 0.89. And that's how you figure out how tall those sum bars are going to be. Now that you have calculated what the tops of your sum bars and bottoms of your sum bars are going to be, go ahead, pause the video, and try graphing it yourself. And when you're ready, continue. Welcome back. Hopefully this is what you drew. So in our conclusion, we would say that there is a statistically significant difference between all 3 groups. Now, these two got really close, but 0.26 is still higher than 0.2, so there is a statistically significant difference between these two, these two, these two. There is a statistical difference between all 3 groups. The rest of this activity we're going to do in class together so that you get practice with drawing sum bars. I hope this video was helpful. Thanks for watching. Bye.